Hi, this is Mohammed Shaaeli, and I'll be talking to you guys today about Dr. Frank Albert, a faculty member at the University of Minnesota, for my biology role model project for Foundation 1951. So Dr. Frank Albert finished his high school in 1997 in Germany, followed by a dual degree in biology and computer science in 2005 from the University of Würzburg, also in Germany. 2010, he received his PhD from the University of Leipzig, Germany. I murdered the name, but I'll put it in the caption. The majority of his time between 2010 and 2016 was spent doing postdoc research and publication. In January of 2016, he started working at the University of Minnesota as an assistant professor at the Department of Genetics, Cell Biology, and Development, a department I want to get my major from. He has his own lab in the MCB building named Albert Lab, and they're studying differences in regulation of the same genome affect the phenotype. Couldn't find any information regarding how he got into science in the first place. He's kind of a private person. Found one interview about him in German, so it didn't help. But it's safe to assume that he was interested in science before attending college because he graduated with a science degree. So, yeah. As far as obstacles, also couldn't find any information, but I think it's safe to assume that there's an assumption. I assumed looks like between 2010 and 2016 he struggled with stable employment. He worked at different places according to his resume. He worked in Germany, United States, Italy, different places in the United States, Italy, China, all over the United States, different research programs. It looks like majority of graduate students struggle with employment for the first years before they find a faculty position. From his Twitter account, I found out that he enjoys biking and mountain biking, an activity that I enjoy too, so that's why I could relate to him. He's a big advocate for science and science policies, science in politics, etc. Also, from his research and previous academic work, I find out that he's interested in neuroscience and computer science, and these topics kind of interest me too, so that's another way I could relate to him. From reading his CBS biography, I realized that both equally fascinated by how complicated gene expression and regulation is and the central dogma in general. So that's one way I could relate to him. Also, we shared similar personality. We both introvert, I think. That's what I read about him. So I admire his work ethics. He's about 35 with 24 publications, so that's a lot of publications for 35. I also admire his creativity and different approach to science and how he combined computer science with biology to come up with a different way to research science. So the majority of his research involves gene regulation as well as gene expression. That's how it relates to my class, to the foundation class. From his CBS bibliography, he's interested in answering questions. How can we tell different DNA differences have phenotypic consequences? What exactly these polymorphisms exert? How exactly do these polymorphisms exert their effects, and how did the genomic diversity evolve? So they're trying to answer these questions from using functional genomics and computational statistical genetics. So the most recent article that was published about my sciences is titled Genetics of Transregulatory Variation in Gene Expression. It was published in July 2018 by an academic journal called eLife. Dr. Frank Albert, his name was the first name on the article, so it means that he contributed the majority of it. The article was investigating heritable variations in gene expression. So to do so, they mapped sections of DNA called EQTLS, which stands for expression quantitative trait loci, and they studied that in 1,000 yeast, over 1,000 yeast. They mapped it. They found hotspots where these sections are found. They found about 102, and some of these influence the expression of more than 1,002. So their experimental method is quite complicated. I read it like 4 or 5 times. I couldn't understand it. According to my understanding, they would grow different types of yeast, and they would extract the DNA, make a library, and then based on that information, map the EQTLs, and then use that information to find hotspots and how they interact with each other. He has a lot of contributions to science, hint at 24 publications. A lot of his research is discovery-based and research finding background information type of deal. One of the most interesting findings of his research is that domestication of a wild animal that's I found really interesting. So they found out the genome of dogs and wolves are extremely similar. What's different about them is that the expression of certain proteins is different, and that's what leads to different behavior. I was referencing the comparison of brain gene expression levels and domesticated in wild animals. One of his articles. He goes in depth in the article and discusses different proteins and all that good stuff. Yeah. So instead of looking at differences, so his research, where he's going with his research, is that instead of looking at differences within the genome, like bases, he's investigating how the rate of expression, how it affects the animal, why is it different, factors, mechanisms, all that plays into it, and how it evolved. So to be specific, with where he's going with his research, he has fundings up through 2022 to research genomic approaches for dissecting regulatory variations. So he's kind of like trying to pioneer or establish a standardized method to approach regulatory variations in the genome, study them, and basically make a database out of them. Yeah, thank you.